So the session that I'm presenting in on Tuesday is looking at stress, stemness, and therapy resistance. And specifically, my talk is really focused on how metabolism can be a target within leukemia stem cells. And so our kind of goal, the overall goal of my lab, is to understand leukemia stem cell biology in order to better target it. And so one of the key questions we asked, and that I'll be talking about in my session, is what are the metabolic differences between leukemia stem cells and our normal hematopoietic stem cells? And we identified that the leukemia stem cells have a higher dependency on these specific metabolites called polyamines. And when you deplete polyamines, we can cause leukemia stem cells, but not our normal hematopoietic stem cells, to differentiate and eventually die. And so we're pretty excited about this, and while we still have a lot more work to do, we think that this is kind of the very beginning steps of potentially a new therapeutic approach for this leukemia. So leukemia stem cells are the cells that are kind of at the root of acute myeloid leukemia, and overall, the therapeutic outcomes of our patients, or the outcomes of our patients, have not dramatically improved over the years. We've seen some minor improvements, and it is a really exciting time to be in leukemia research because there have been a lot of new therapies approved in the last 5 years or so. So I think there's going to be a lot of changes. But right now, we still have a major problem of therapy resistance and relapse. And the kind of central premise of my group is that this is because of our inability to target these leukemic stem cells. And to the best of our knowledge, nobody to date had really understood at the level of individual metabolites what was different, taking an unbiased approach between a leukemia stem cell and a normal hematopoietic stem cell. And so we really wanted to try to fill that knowledge gap. So in this study, what we did was isolate leukemia stem cells from 18 primary AML patient specimens and normal hematopoietic stem cells from 5 normal bone marrow specimens. And then we ran mass spectrometry-based metabolomics to quantify the different metabolites that exist within leukemia stem cells compared to our normal stem cells. And this is something that people hadn't done before. So in the field, people had taken kind of biased approaches, looking at very specific metabolic pathways, but we really wanted to take a completely unbiased analysis of metabolites within these two populations. And what that enabled us to find was these specific metabolites called polyamines, which hadn't been described really in leukemia stem cell biology before. And we found that these metabolites are really important to control stemness and differentiation. And importantly, when we deplete these polyamines, we can cause leukemia stem cells to differentiate, but it does not affect our normal hematopoietic stem cells. And we hope that this is kind of the foundation of a future clinical approach that we could take to target leukemia stem cells. For me, as a kind of more basic science researcher, once we have really kind of exhausted what we can do in the lab, which is a lot of kind of preclinical patient-derived xenograft modeling, which is what the bread and butter of my lab is, we would look to partner with physicians and physician scientists to kind of initiate clinical trials, which is something that I've been fortunate enough to do in the past, and I hope that we can continue on doing that, and potentially even for this project if the data continues to look good in the preclinical models.